E intrat în bucătărie. Ok, deci nu trebuie bine. E live? Da, e negru, e ok, că am pus eu negru. Și ia, nu vorbiți. Acum dacă dau sin. Gata. Bun. Gata, am ceva până la... N-am făcut că doar e pregătit. Da, liniștită. Luați-vă de la tipii care fac cafea, nu vă luați de la aparat. Ok. Ok, bun. N-ai pentru ce?
Am descoperit ceva rețele africane sau droguri care vindeau la școli. Lângă școli, droguri. Întâlniri.
Mai greu. Așa am făcut și eu. Asta
Tu îți faci toca pe lips. Îți faci ceva pe tocă.
Dacă fac eu singur, poate rog cuiva să-mi facă ceva.
Da, mi s-a părut să mă pune.
Deja am făcut să mă fac, dar nu mai am...
Eu mi-aș pune părpurile de la PDL, din așa dus cu resilience.
Aș pune un mic, da.
Wow, și aia atât tare.
Am făcut ceva la Catering, desigur.
Nu știu dacă sunt ușor. Mie mi-ar place să pun ceva stretchat foarte meaningful, dar în același timp. Mă, rest, deja mi-am făcut din tocă ceva. Gen,
Tu singură?
E fain.
Mișto.
O să faci ce alea atunci?
nu, conspirații. Conspirații. Am vrut neapărat ceva pe temă de space și poate sus ceva pe temă de business. Cu marcări acrilice am testat. Pictat, desenat.
E bună cu LeBron? LeBron mai sunshine. Completarea galaxiei.
Uite.
Fain rău.
Cristălițe. Datele de mine de sus.
Deci eu e ultima. E ultima până la urmă?
Ce?
E tu o să fii ultima până la urmă? Draw sau draw?
Sau.
Draw.
Sau.
Deci că trebuie.
Degetă.
Iolul? Bulgarii?
Da.
De
fapt, Fante de Vasole. Nu, Fante de Vasole este. Comună.
Patriolul, Bulgarii.
Faseole în bulgarii.
Faseolă în bulgară.
Păi e fasole comună. Da, e plantă de fasole comună. Salut! Bine. Eu o să mă duc în studio. Nu ar trebui să fie
Da, vine cineva de la voi.
din
Echipă. Am pic frumos aici. Cam așa, da.
Mulțumesc mult. Mă auziți încă? Ok, vreți să pun ecranul cu prezentarea titlul sau fără? Deja, sau să fac stop share sau e bine așa?
Puteți sta cu titlul și cu prezentare.
Deja, acum este?
Da.
Ok. Ok, e mai bine așa? Ok, tot așa.
Dacă nu poate da full screen, sau că în partea nu se vede.
Oare puteți da full screen să cum acest slide?
Slide-ul acum?
Da.
Eu nu am ajuns, nu mai văd să nu.
Să lase cum a fost.
Să vedem numai ecranul, nu știu ce să vedem acolo. Eu nu văd de prezentare.
Să vedem exact modul ăla de prezentare pe acel slide ăsta. O puteți lăsa cum e cu doar cu
Dar să vede suficient mărimea ecranului, așa.
încercăm noi acum să vedem cum dau un pic mai mare.
Să fac eu o să
Hai, ok. Bun. gadać.
Też powinna być.
No, wystarczy szafę.
Jakaś do tego wieku? Dominik? No, był.
See everybody in the seats. We hope you enjoyed the lunch break, everyone. My name is Felix, and I will be your moderator for the next two sessions. Next up, Sofia Pascu, who will be joining us online, as you can see, will take us through investigations into organic, inorganic hybrids as fluorescent theranostic systems, confocal imaging, and NPFLIM cellular mapping. Sofia is a professor of bioinorganic and materials chemistry at the University of Bath. Her research involves working at the interface between organic, physical, inorganic, and supramolecular chemistry towards applications in sustainable chemistry, Fischer-Tropsch catalysis, CO2 storage and conversion, dye-synthesized solar cells, metals, and in medicine and nanomedicine, and multimodal molecular imaging. Thank you for participating, Sofia, and we are excited to hear about your work.
Thank you very much for your kind introduction. I wonder whether you can hear me well. I've already shared my screen, so I think probably you cannot see me or only see me as a tiny, tiny picture on the screen. I'm delighted to be there, though. Sadly, I cannot be in person today. I've got 4 more hours to teach before the semester finishes all today. And so I am there in spirit, and I miss you all very much. So I'll take you through a few of my recent results, which were obtained by my PhD student, Megan Green, who finished a couple of months ago, and previously a couple of other PhD students, one of whom now works at the Memorial Sloan Kettering Cancer Center in New York, and the other one who is still Rudy Rekner, and the other former PhD student who is now a postdoc still in life sciences at Bath. His name is Howard G. He did a lot of the cellular work. I was asked to reflect perhaps a little bit on the importance of supporting students. I'm deeply passionate about working with young people, working with students. I still feel a student myself. We're all learning together, and I'm very much interested in what happens after once they finish their masters, they finish their PhD with me. What is their career progression? I am also a grandmother. Some of them already had children of their own, a scientific grandmother, and they are sending me the photos of their family life as well as their career, and I'm very, very much excited about them. They have about 6 or 7 grandchildren, and some of them have met in my group and married and gone into the center really happily, and I'm still in touch, and we still publish together at times. This left me, essentially, overall, my work revolves at the interface between molecules and materials, looking at applications, looking at interesting things molecules and materials do when they're assembled, and essentially the wider role is because of chemistry, but also the university and the UK scene and worldwide is now defining sustainable processes as strategic needs, which is understandable in a bigger context. So we all align to the various aspects of sustainability, systems and systems processes, including systems chemistry, and a lot of the things that we're interested and passionate about in the group are supposed to bring us closer to applications. Here are some of the general things that we have are published over the years. So today I'm going to look at one of the teams that I'm working with, looking at, as a chemist, design protocols, tools, assembling multimodality products that have got a role in bioinstitute application, and particularly using fluorescence imaging and as an investigation tool, especially deeply into biophysics a little bit. There are closer aligned projects in my group which look at drug delivery systems, also some nano-encapsulation in general terms for a range of purposes. But generally, we always see in new applications for functional small molecules and materials looking towards medicinal arena. But remembering that we are chemists, and dipping a little bit into life sciences is actually exciting, but it doesn't mean that we have all the answers to that sort of viewpoint. And so, in terms of the scope of our work, really, we're looking at ways in which our molecules address nuclear polymodality, nuclear imaging applications, and looking at the near-IR emissive fluorescent probes for more detailed information about the mechanistic understanding of processing underlying health and disease, healthy and disease systems across in vivo, in vitro, but also ex vivo in spheroids and in organoids and as well. Considering applications in nuclear medicine, there are a lot of challenges that need to be addressed before that becomes an accessible diagnostic method in medicinal approaches worldwide, especially lack of infrastructure and cost. The fluorescence imaging, optical imaging in general, was much more widely available, but there are challenges in terms of availability of near-foldable probes, which are actually active in the near-infrared region, because of course such systems are suffering from the interference from native biomolecules. You can see here in this diagram, which comes from the Chemistry of Molecular Imaging reviews, like here. This is a well-accepted diagram where we can see that we need to avoid the overlap with the fluorescence of biological molecules if we were to achieve the ambitions of optical imaging. So moving from visible towards far red and near IR would give us the spatial resolution that we need, depth in terms of the type of tissue that we can access, which currently is smaller than 1 centimeter, so that isn't particularly helpful unless you're looking at biopsies. Key use currently by the visualization of cells. So the complementary techniques to nuclear medicine advances, which as you could see have a lot more relevance in metabolic imaging. The optical imaging techniques have quite a GDSR potential, but mainly because there are sufficient chemicals available to address those challenges. And so, particularly in terms of radiochemistry, radiolabeled drugs that are out there available for prostate cancer detection and treatment, which is one of the case studies that goes on in my group. There are a number of systems both available in diagnostic settings, as you could see here, and they all rely on small biomolecular targets, but also some that are already in treatment in the radiotherapy perspective. And so the combination of simultaneous diagnosis and therapy with the same molecular probe is what is now considered to be the future of the theranostics as a field. And so these are the radiolabeled medicinal probes in various stages of clinical trials and clinical use, particularly looking at some gallium compounds that are radioactive as metalloprobe. And as an inorganic chemist, I'm much more interested in what metal coordination compounds do. However, the more accessible ones are these shown here, the fluorine 18 and FDG particularly, that is the sort of workhorse of imaging for diagnosis purposes, including in prostate cancer. And the very recent one is PSMA Lucas-Vera compound. So the full peptide, translating linker, traceable linker, small amount of fluorescence, and the radiolabels to tissue, and this is the counterpart treatment for the diagnostic gallium-related compound. In terms of the relevance of the message process, the cancer cells are available to us through a wider program with the Royal United Hospital at Bath, and the addressing imaging and therapy in prostate cancers is, as you can see from this graph here, quite a relevant one. It's one of the ones that in the UK at least is deemed as one of the most at risk. In terms of how we look at the approach in my group, we're looking at repurposing tyrosine which are derived from tryptine, which is a well-known chemotherapeutic, and conjugating these with molecular tags which are fluorescent. In terms of the state-of-the-art, the literature available there, essentially there are a number of tryptine-inspired tyrosine kinase that have been studied, and we know they have potential to interact with them and also show high toxicity. And so recently we zoomed into these two ligands here, HF1 and HF2 shown here, and these we functionalized the number of metal complexes. The HF1 is new. HF2 has actually been studied and arrived at some way further down the line in clinical trials in early 2000s. But of course, there was, from my perspective, can bind metal, are metal complexes, therefore they're something. And so conjugating these with what APA dies, this is again the state of the art in metal complexes with various organic fluorophores, including those that push position towards the near-ionized. I showed here they have been reviewed. So overall, Megan, my student, looked at bringing together some aspects in which fragments of a complex prostate cancer targeting group that is overexpressed on the surface of prostate cancer cells, shown here together with a range of fluorophores, and conjugated to tyrosine kinases which can bind metals to facilitate translocation to allow us to deep into the concept of metallomics, and those metals can be radioactive. So this is generally the overall design elements that were enjoyed. In terms of how we make these peptides, in my group we make these peptides on a laboratory scale rather than an analytical scale, using microwave technology, which is a sustainable way of looking at it if you know the fact that we're going through liters of BMS, which is sustainable. So that hasn't yet been addressed. However, the amount of peptides that we are making is about 250 milligrams in one go. This allows us to dissolve synthesis approaches, allows purification, characterization, and beautiful NMR spectroscopy on rather complex molecules. Because there's this fragment of the monocytic peptide, which is known to target the adherent receptors found in abundance in prostate cancer cells. Were able to then take further for synthetic purposes, which I will show you a little bit more a little bit later. In terms of the functionalization of the priority-like monotherapy carbon, their reactions are straightforward to temperature quite often, and they give rise, depending on the nature of the metals, we look at gallium, copper, platinum, as well as with various stoichiometry. For gallium and indium, it seems very much that the preference is towards this distorted octahedral type structures. With platinum and copper, it varies quite a bit. We are able to characterize this very simple system. Actually, a bachelor student in my lab has now just made a lot of these variations of this metal complex and with some crystals which she analyzed as a perfect type of system for starting with PhD and then complicating much later over doing masters and PhD level work in. In terms of how they interact with these cancer cells, we can see that with gallium and indium compounds, with both ligand 1 and 2, are actually very active. They compare very well with what we expect from cisplatin in the same sort of conditions. In fact, cisplatin is slightly less active than these things, which is good news in some ways, bad news in some other ways, because we don't really want to dispense poison. We want to make them therapeutic. And so these systems are clearly very potent, and even more so when a metal complex is involved. And so we then proceeded to functionalize these with these ligands with a Modifi alien conjugation here in the root. So we're making our own Modifi, we don't buy it. We make it on hundreds of milligram scale. Typically in over a couple of days you could make yourself a large amount of ready to use precursor, and then simple reflux in acetonitrile gives a pyrophenic acetone that essentially has exchanged, has this NH2 group here for the primary amine that we started with. And so that gives us in rather low yield after a lot of flash chromatography, the desired ligands shown here, and in terms of formation of metal complexes, that is then quite straightforward after this very simple reaction. Gives rise to indium, gallium, copper, platinum compounds. These for indium and gallium, we have two of these units binding, and the camera is PS6. Of course, indium and gallium are pretty flat, strongly standing, and they have very interesting behavior in solution, which we will have time to go into. The copper is a little bit more, little bit less behaved. It gives some very nice, as expected, flavor compounds. In terms of the crystal structures that we have available, we have characterized these from the silicon to the metal complexes. We also grew crystals of the Modifi NHS, which turned out to be quite stable as an intermediate, and we formed the desired ligand, which we then characterized by crystallography, which we're very proud of, because it was actually not that simple to go down crystal. We just had this paper accepted. Actually, it's now online as of a couple of days ago in Advanced Science, which we're very pleased about. And so for the ligands involved, we figured that actually we need for cellular transportation maybe a little bit more modulation of uptake. So we transformed that pyrophenic acetone framework into a pegylated version, again using this Modifi and Modifi NHS as a starting point, slightly different method than before, and we were able to get the platinum compound, which I should have said that for all existence we are able to do a quantum yield determination. We did this in methanol, but we also now reported it in DMSO and in DMSO PBS mixtures. In terms of the emission excitation mapping, we can see that even with copper, we still retain some of the fluorescence, albeit a lot weaker than from the original ligand. But we can still see that it's traceable, it's going to be traceable in cells. We were pleased to see that for copper we still retain very high activity that was seen before. So that is very much convincing that we didn't really destroy the thermognostic potential by including the optical traceable body together with a metallo carbon. We also saw that for this copper compound, we do have some release of the copper, which is, of course, evident then from the enhanced fluorescence emission. When we just have the free ligand, the fluorescence would be higher than maybe quenched by the D9 copper 2 with 1 ampere electron that's bound there. So here's the fluorescence monitoring of this. So in terms of cellular uptake, what we see here is that the ligands themselves, whether it's the enzyme linker or with the more complex pegylated linker, are all taken in, just the same going into the cytoplasm, going into the endoplasmic reticulum, very much what dictates what goes on here for the free ligand. That's a very large molecule, and it's known to localize in the endoplasmic reticulum and also degree of very good modifical properties. And so we have all of the localization studies that were carried out within. In terms of the uptake, we expect an active form of uptake, which is the average at 37 degrees, showing good cell penetration, and at 4 degrees, showing less good cell penetration. And this is when we excite with our laser and foreign laser nanometers, and we pick up the emission characteristics for the body in the green channel in the middle, where there is density in a blue emission or red emission channel. We can see here that the PT3 cells are pretty much expected as a range of not so pretty cells, and this is characteristic for PT3, which is not a great cell to work with. It's a little bit hard for my students to work with it, but this is the one that the medical team is interested in. And so when we attached indium or platinum to the systems, we saw subtle changes. So I said indium is a strongly classic, and for that we actually were able to see some of the truncated regions in there. So the presence of indium does change somewhat the localization. There's more things being taken into the lysosomes, and for platinum likewise. What we see with platinum is a little bit more of a cell damage. Some very small amount of the nuclear uptake, but not convincing. In terms of the in vivo testing, we took the gallium compound, the indium counterpart, and we saw that gallium does slightly different localization in this living zebrafish. We're looking at some structures that are unique. Neuromass can be labeled with this fluorescent value and carbon, but overall everything can be into the gut. So we're now trying to understand a little bit more about why indium versus gallium behaviorally on the timescale of an hour tracing in vivo. Sorry, I don't want to do that. Because we were interested in how this damage cells, we also wanted to know what happens in isolated nuclei and isolated chromosomes. And so here we see a fluorescence lifetime assay on a isolated nucleus with corresponding chromosomes as well as the chromatin molecular nuclear material. And this indium compound seems to sustain the cellular membrane quite well, and also when the nucleoli as well as the isolated chromosomes are available, they seem to pack in there. So that is characterized by a longer-lived fluorescence lifetime as opposed to the shorter-lived lifetime shown in green. So the longer-lived lifetime of the compound, which indicates association with chromosomes, actually seems to indicate that the compound is aggregated there, it's packed and immobilized. And so we see here some of the parameters in terms of the shorter-lived picosecond level as well as the longer-lived 2.2 nanosecond level, and the mapping corresponding here to fluorescence mapping in fluorescence lifetime in the rainbow color, and then the fluorescence emission next to it. No color particularly. This is done at 48 nanometers excitation. For the indium compound, for the platinum compound, in isolated nucleoli and isolated nucleus, we see an even more interesting picture because actually in fluorescence lifetime we see these things aggregated in the nuclear material. But in phosphorescence lifetime, which we still decompositing, actually we saw the presence of platinum together with the ligands associated in the same spots as indicated here in blue. So this is very much work in progress. We're very, very excited about these results. We don't know exactly fully what they fully mean in the context. It's just that it's clear that our platinum compounds also, similarly to the indium compounds, like to go and bind to the nuclear components, such as nucleobases in the isolated chromosomes. And so, inspired by the radiochemistry work with the targeting peptides to change drugs, we were looking at the semipartisans and their conjugation with the peptides. And so, particularly from a perspective of a longer idea in target hypoxia, both with copper-related semipartisans, with gallium-related semipartisans. We reviewed the field a few years ago, maybe it's time to do it again. And so we had some success earlier on already then university with looking at alternative gallium compounds that were radioactive, and we showed that there is some hypoxic uptake. So this is in cells and tissues which were deprived of oxygen for these. However, we didn't see any particular tracing and correlating with the prostate cancer tumors for these, possibly because these systems are in fact excreted a little bit too fast, which is just a good thing, but they, as you can see here, is mice work that at 30 minutes, the compound, the gallium compound shown here, has been able to localize in the bladder, and then off it goes with clearance and then remains trapped in the lung. So we didn't really think that that was as standalone small molecular compound going to be sufficiently selective, and the thing that we are working on is essentially ways of functionalizing these with a range of these peptides which we know target PC3 cells preferentially. So we have made the structural and peptide sulfuric acid test characterized by the mass spectrometry and HPLC. Further synthesis work monitored by HPLC essentially allows us to make the compounds with zinc derivative that we know is biocompatible, so either with ethyl or allyl, which is identified to the mobility conjugate, the logic 3 and 4 here in this slide. And that then allows us to monitor the system in cells, again by confocal fluorescence, as well as by fluorescence lifetime, which is in two-photon mode. This allows us to get rid of the interference from the autofluorescence and other biological materials that we're looking at. So we're looking at 910 nanometer excitation, moving towards treatment with two-photon approaches towards further red. So these targeted metalloprobes, the compound 2, I believe, with ethyl group here, which has got the right balance of hydrophobicity and hydrophilicity, seems to work really beautifully in recognizing breast cancer cell membrane, which also overexpress the GIP receptor, the FGF7. Prostate cancer cells, PC3, they are taken up very, very well, even inducing some shedding and destruction of the membrane. But interestingly, they also go into cervical cancer cells, which do not have the membrane receptors in the same high numbers. So that means that again, my memocomplex dominates the uptake, and so it's probably a constant mechanism by which these things go through, not just simply membrane recognition through the peptide delivery. And so this is what we're working on at the moment, trying to study in a range of systems, including isolating organelles and including liposome treatment to see how the interaction with the range of lipids occurs on these types of compounds. And so we moved swiftly. I think I'm running out of time. I don't know exactly what the timeline is, but maybe somebody can tell me when I'm here. So to move further in the NIR, we actually looked at using this type 7 fluorophore as functionalized with a peptide that we looked at. So we have Limfoclu, which is also one of the peptides that is used in the theranostic system that is Lutathera, as I mentioned, also our organic fragments. And because we have all these peptides available in large scale, the challenge then became how do we get hold of these dyes that is actually very expensive to buy from individuals and also do chemistry in a large scale to bring together the fluorophore radioisotopes and the delivery vector. This type of family of near-IR dyes is now established in preclinical trials for a range of things, and so while we can buy some of the courses, cost us prohibitively, because that's totally not sustainable when we're starting to look at synthetic chemistry. So we reviewed the areas, what is available there by means of synthetic chemistry and structural chemistry with particular sulfates a while ago. And so we adapted the synthetic protocol to make quite a lot of these compounds. This is the work of Rüdiger Exner, who is a fantastic organic chemist, who in my lab, supervised by an inorganic chemist, was able to make hundreds of milligrams of this expensive near-IR dye, and with a range of linkers available, but we have to use microwave reactions to reduce the reaction times, which avoid cyclization inside products. We're able to characterize coursers by NMR spectroscopy, and for this you need milligram cell size and samples, which is impossible to buy. A dosing spectroscopy shows that pure compound, pure HPLC, so we're kind of confident that we have what we need there, and we're able to do a lot of the fluorescence lifetime spectroscopy, sorry, getting ourselves ready for lifetime imaging with these things. In terms of the photophysical characterization, we saw minimal aggregation and concentration needed for work, and the expected parameters are in line with commercial or related samples. You could that we were after. In terms of the cellular uptake, we needed to go to near IR. We needed to excite the 780 nanometers that required building some lasers and test systems to get. Interestingly, what we see here is some really nice displacable uptake and some differential uptake with respect to healthy cells for the CP3 versus CHO cells. In terms of the lifetime imaging, we have all the correct parameters comparing up here the parameters in fluorescence mapping of the dying cancer cells with respect to the solution spectroscopy. So these are maintained in these boxes. This is a droplet which we see inside cells. The compound remains intact, which is what we after really. And so we went and functionalized this with CI4, cell penetrating nuclear, natural product, and then characterized these compounds fully. We also introduced the zinc and compared the zirconium 89 radioactive incorporation with the well-known GF4 chelator. This is a compound well-known and understood in zerophores. And so, but for these, we have to work with a colleague in Cardiff in a medical school and use their protocol for the zirconium 89 labeling. Which we then compared with the GF4 bombazine uptake. This really worked very, very well in terms of the radioelectric of peptides. And then we moved to use this transmethylation protocol from zinc to zirconium 4+, and we monitored this by radio PLC. Then we looked at our own interesting sulfate DFO compound, and again we see here the uptake beautifully compared to the free zirconium, which appears in terms of the radio PLC parameters in an entirely different range. We are satisfied that we have very, very good more than 90% incorporation. So this is now awaiting some in vivo work for imaging side of things. And we monitor this by voltage, radio HPLC, which is hard to do with this compound. A radio PLC would be easier. And so other compounds that we looked at are essentially the Scopulu, which is an anti-PSMA agent we introduced that to the carboxylic group. We also looked at the uptake in cells, which was to us pretty successful, and I didn't really think that this would really go through. But again, the sulfate does drive the uptake of the entire probe. So overall, we looked at incorporation of CF4, incorporation of bombazine, incorporation of PSMA through a range of methods, both in chlorine sites for the CF4 and at the carboxylic group for the peptide. And we looked at the radio labeling again for the DFO as a conclusion. But for us, the monitoring by fluorescence lifetime imaging, especially for these bombazine conjugates, is what drove the process, because we understood that there is a need to more subtly compare and contrast the uptake of the free sulfate with the peptide conjugated sulfate, and the confocal fluorescence alone in emission mode just shown here in black and white, it cannot be parameterized. It's not sufficiently analytically informative to show where the distribution of the compound is if there's some thermal damage to the probe. So here we see, and then rainbow color say that I have a pretty narrow distribution. This probe doesn't get particularly damaged. So I would see a very, very different profile if the fluorophorin wouldn't be intact anymore. And so this is where we are with this. I left plenty of time for discussions in case there are questions from the audience. In conclusion, we had looked at the large scale synthesis of the dyes and peptide conjugates which we have available in over 100 milligram scale. We typically use microwave irradiation or room temperature reactions because for tyrosine hydrogen especially, cyclization is a problem and then it's nearly impossible to separate these compounds. Also for sensitive dyes such as cyclase, damage through high temperature and prolonged temperature control reactions isn't at all good. So for complexity of peptides, work with dyes and kind of complexes, we found microwave irradiation is best. We working with the research complex of Harwell, which is near the Sixtron big laser complex funded by NCSC to look at the fluorescence lifetime in near-IR detection mode, because that would be a better, more informative tool in terms of understanding the optical properties of a library of cyclase fluorophores. They compounds, so we're not just looking at emission in fluorescence emission, but fluorescence lifetime in correlated modes to same field of view. Radio labeling is hard to do, and particularly because in the UK we can't really get access to metalloisotopes everywhere. There are a few collaborating groups that can provide these. But for that, it meant that we needed to develop assets that are readily deployable and to be very agile in the way we access this facility. So using translation assets, often from pink intermediates to introduce gallium or zirconium into framework. We're looking at hypoxia targeting as a direction of travel. These are a very, very interesting systems. Hypoxia occurs often in large tumors, but it also is relevant in stroke or processes that are linked with deregulated wound healing of relevance to diabetes. So there's more there that can be explored in terms of the role of cell lines in hypoxia targeting. We also work with modifying Cy5 DFO conjugate. So this is therefore is very good at cell penetrating, but it's also very good at conjugated metals. Cy5 is a little bit easier to sort and name than Cy7. So Cy6 is something of cyclase are very, very interesting. Needs good organic chemists. This is where we are. The Zirconium 89 will need more optimization work and some artists might. Radio HPLC is currently difficult because the zirconium is very strongly SASP2 and it's very, very oxophilic, and so it sticks on various solvents, and it sticks on the board with HPLC. The C18 columns are not that great, and so this is me with it reminds me to thank my chemists and biochemists. They start as chemists, inorganic chemists, and somehow for 3 years drive more the chemistry department in the UK. However, this is as helpful as still in my group. They two have just completed their PhDs and we're writing some of their papers. Megan is the first author of my advanced science paper that just came out. It summarizes most of this work, and Rudiger has maybe still collaborating closely on the Coding-89 work that I take. And of course, the importance of collaborators, mentors, and friends is huge. I have been very, very fortunate to be able to work with some amazing people, and I am very much hoping to continue to strengthen my links with Cluj. I want to be back. I miss it terribly, and so thank you for the funders, and thank you for all of you for your listening, for all of you for your listening, and giving me an opportunity to be at home, even online. Mit Melfalten Hult.
Thank you for your engaging presentation, Sophia. Now, time for questions, please. Okay, well,
I have
oh, please.
malo doară.
Se aude? Do you hear our question?
My name is translate.
Poți să te duci la...
I need to say it louder.
One second. You come here.
Yes. Thank you for your presentation. I have a question about the compound in the last part. You showed a dose experiment, and I was wondering what was the purpose of it in that case?
Let's see if I can share again. So the purpose is that typically purification is hard. Let's see if I can find this. I know what you mean. So purification for these compounds is incredibly hard. They stick on the 18 columns, and so you need a lot of these flash chromatography experiments to get the compound as pure. The physicochemical properties of the final product, that of the oxo substituted variant and intermediates are actually quite similar. And so you can't put into cancer cells and expect to see phenomena, compounds that are not pure by more than 90-95% level. And because you might see the toxicity that is due to some other components that's very potent. So this is one of the most challenging molecules to make, even though it's commercial. You can buy it in micromolar concentration in a vial. You can't do chemistry with the nano. Well understood and not available, so this is why I'm putting in. The dosage spectroscopy shows us, and this is done for all the compounds that we have. Really further evidence of the purity and dosage spectroscopy is seen for the Modifi compound in my paper that I just showed off with earlier. Really, I don't think I put dosage for the rest, but dosage is the one way of being sure that you only have one migrating species there. Yes, so for all of these ligands, especially the Modifi characterized systems and the metal complexes, you need to characterize those by dosage spectroscopy to make sure that the very large compound that you're after, I don't think I put it here, the very large compound that you're after is there and more than 90% purity. So for more information as to how we use this and why, I'm very proud of this because actually I did a lot of DNA PhD work myself for the students, and it involves 3 or 4 of my PhD students that work over quite a few years
now, are there any other questions from the audience? Okay, in that case, Sophia, I would like to thank you again for your presentation and enjoy the rest of your day.
Thank you very much. See you all soon, hopefully in the summer. Bye.
Now we invite you to take another coffee break, the last one of the day. Afterwards, we will have our last presentation, oral presentations today. We start at 20 to 3, so in around 25 minutes each.
Ela é um pouquinho negro.
No.
Okay, welcome back everyone. We hope you enjoyed your break. For those of you who weren't here the last session, my name is Felix and I will be your moderator for this, the last session that we have today. Can you use the mic? Okay. I'm sorry. So
This chemistry session is comprised of 5 oral presentations. Good luck to the presenters, and to the public, don't hesitate to ask them about any curiosity you might have. First off, we start with Lucia Alexandra Grădinaru with computational approach to explore bilirubin-like compounds.
Hello everyone, my name is Lucia and today I present my slide computational approach to bilirubin-like compounds. Based on scientific studies, bilirubin is an endogenous compound produced by oxidative breakdown of them, and at high concentrations the attachment to the toxicity may influence the inflammation processes. Our objective was to identify bilirubin-like compounds that possess the issue of low solubility and high risk of toxicity while keeping their therapeutic effects by evaluating their anti-inflammatory potential in targeted regulators of the nuclear factor kappa pathway. To complete our objective, we analyzed three hydrolysis, IgG alpha and uARF thyroid kinase, and JAK2 to assess their off-target activity. The computational methodology was structured around 5 key approaches, including pharmacogeneration, the characterization of ADA box and reactivity, and also molecular looking and chemical space analysis. Moving on, but the results section, we can observe how bilirubin interacts with the human serum albumin proteins. These analyses reveal several components, including hydrogen, hydrophobic and aromatics interactions with the specific amino acids in the active site. These analyses reveal several key components, including Okay, based on these interactions, we generate a pharmacophore model, Pharmate, which identifies the future compounds for bilirubin activities. Using this model, 9 promising candidates were selected from this databases for further analysis. Then shaping was used to analyze the pharmacokinetic properties of the selected compounds. From the bioavailability table, only compound 1 is highly optimal, while bilirubin and compound 2 show the most pronounced bioavailability limitations with poor properties outside this optimal range. The Boydek plot from Swiss AD illustrates the predicted gastrointestinal absorption with 4 compounds fitted within the white areas with high gastrointestinal absorption, and the remaining bilirubin fit outside this white area, suggesting lower gastrointestinal absorption. As you can see, none of these compounds fit in the lower area, which means that none of them are crossing the hematoencefalic barrier. The toxic profile of bilirubin and selected compounds were performed based on Osiris Protein Explorer. Osiris provided insight into solubility, toxicity, and drug-like exposure. Some compared to bilirubin, most compounds show greater stability and higher drug-like scores. As you can see, a few were left behind, but most compounds were deleted. After this test, from 9 compounds, we obtained 5 promising candidates to the next, the pharmacochemical characterization of these selected compounds were performed using the Schrödinger assay, including hybrid functional parameter combined with standard system. By analyzing the molecular results below, we can observe how the electron density is distributed in bilirubin and ethanols. The color maps indicate electron accepting and electron donating regions and highlight significant differences in compounds reactivity and stability. And the energy gap was calculated using the homologo-valued energy. So the smallest band gap among the compounds was for compound 1, and all three compounds show a higher band gap energy than bilirubin, indicating the greater relative stability. While compounds 4, 5, 6, and 8 show low electronegativity values, compound 1 presents a higher index close to bilirubin, and which means suggesting similar electronegativity pattern. The DFT characterization was followed up by molecular tracking analysis, which began with protocol validation. This includes reducing the comprised elements into their structures, achieving the maintenance verification test below the separation of those two arrangements. After validating the tracking protocol, the doctor selected all the selected compounds with the light standard precision mode against of all the three classes. As you can see, compound 1 achieved the more favorable light score than the native ligand of 3RZF structure against IKK target, and had a similar score to the native ligand for IKK alpha isoform. Looking at JEC2, we can see that the compound 1 doesn't behave well at all, with very low binding affinity, almost a half to the JEC2 native ligand. And the MHGSA calculations are consistent with those obtained from GRIDE, indicate favorable results for compound 1 against IKK alpha and beta kinesis, and completely unfavorable results for JEC2. In the next step, we analyze the compound 1 in relation with the results obtained from GRIDE and the results of the fluid reactivity analysis. The arrows display the highest function index corresponds precisely to the hydrogen bond acceptor errors identified in molecular tracking analysis. Confirming its role as potential inhibitors. In addition, for this compound, we analyzed the chemical states in relation with the IKK alpha inhibitors, beta inhibitors. We performed physical component analysis based on physical characteristics and apart from molecular mechanisms. Compound 1 relies on IKX very close to the F compounds, which are too potent and experimentally validated inhibitors. So, in conclusion, our integrated statistical approach enabled identification of different compounds with improved analyzed properties, with compound 1 emerging as the most promising candidate, since it also fits in the known IKK inhibitors. However, experimental validation is
when you need to scientists remains essential to confirm its potential therapeutic effect. And here is our progress on the first step pharmacomodeling to the last one presentation in self-testing analysis with molecular inspiration in progress. And the session plan is to publish our results once all our questions are covered. Thank you.
Great presentation, and thank you. Lucia will now take your questions. Yes?
Can you go into a bit more detail into how you theoretically predicted the toxicity of these compounds? You mentioned that on one of your earlier slides.
Yes, I mentioned online course or series. Sorry. This tool generates automatically these results.
And what is it based on? These are toxicities.
Based on the structure.
Okay. And does it compare it to active sites in various, does it have like a list of potential sites that influence its toxicity and then it compares the structure to what could fit in those, or how does that work?
Sorry, but I don't think that this question the details of the program. I'm sorry.
Okay, I guess you could discuss it afterwards to clarify. Any other questions? Okay, in that case, thank you very much again. And please, another round. We move on to Razvan Yuri, who will present the synthesis of 2,2'-bipiridi, 6,3-mercury 2 chloride, and transmethylation reactions. Good luck.
Thank you for the presentation. My name is Razvan Yuri. I'm a third-year student at the Faculty of Chemistry and Chemical Engineering here in Cluj. Even though I'm an engineering student, I want to present you a chemistry topic. I'm generally working on organometallic compounds and their transmethylation reactions with different P-block metals. So, since like 1 year and a half, and I want to present you a part of my past research in the organometallic field. So, we started thinking about this proligin. I don't know if you can see the pointer. It's okay. So we started with this proligin, which was already known since decades, and we observed different. I gave only 2 examples. There are more different metallic complexes using transition metals such as gold and palladium. And in another case of a proligin, we found one with rhodium. However, we wanted to, because in our work we work with main group metals, we wanted to see if we can synthesize some other derivatives using P-block metals. This was from the presentation from last year, but I will revise it a bit. This is the general synthesis of tetraproligin. We take 2,2'-pyridine, and in nucleophilic addition we can lithium in DHF. And then hydrolysis and then oxidation with potassium permanganate and the chromatography, we can obtain this proligin, which was noted PPPH hydrogen, and we have picked a constant notation during this presentation. And then, in reaction with mercury acetate, in boiling methanol for 3 days, and then adding lithium chloride and quenching with water, we could obtain our organometallic compound number 1. This was already reported in literature a long time ago, like 1990. but they mentioned with 15 to 20% yield, and we could obtain at around 45%. And then we obtained an organobismuth complex by the reaction of the organometal with bismuth trichloride, as I was saying, a 3 class for around 18 hours. So we could obtain it as a white solid, insoluble in dioxide. Since last year, more stuff happened, so we could obtain other derivatives with high valence metals from the P block, such as antimony, tellurium, tin, lead, germanium. I will not present all of them today, so if you have any further questions about any of the others, you can always ask me after the presentation. But I will focus on the organotellurium-4 and organotin-4 compounds. So, this was the first struggle when we did this synthesis. When we try to obtain the organomercury compound, we also have a coordinated compound like this, which wasn't. Yeah, this is a bit of my mistake. It should have been an acetate here. And we have to separate the two compounds, the organometallic one and the coordinated one. We are boiling in ethanol and filtering while it's still boiling. And the insoluble part will be compound number 1. Then for this, we fully characterize it by NMR. The protons will be presented a bit later, but here is the mercury NMR. At around, we could see a singlet at around minus 1030 ppm, which corresponds to the region in which we expect to have the signal. Next is the general synthesis. So here are the two syntheses that I will present today, but we try to use the same conditions for all of the complexes we synthesize. So the organometallic derivative is dissolved in dioxane together with the high valence metal chloride or acetate, acetone for lead 4. And we let it for reclass at around 18 to 24 hours. And then we on-filter the solution and wash with dye filter. For the organolead 4, we have to also add chlorhydric acid in dilute and let it react for 2 hours. And we could obtain all of the compounds obtained as colored or white solids, which are insoluble in the reaction mixture. For compound number 2, the organotin-1, we expect to have 11 signals, all of them in the aromatic region, which we could be seen here. We measured in DMSO, this is DMSO. And we could see all the 11 signals in the aromatic region, together with this signal for proton number 1, for which we could also see that satellites correspond to the TP. Then we also measured at 119 tin NMR, and we could see a signal at around minus 450, which also corresponds to the region for organotin-4 compounds. We also measured carbon and bidimensional NMRs, and for this, from the reaction mixture, after the filtration, we could obtain some crystals. And we get this structure, a single crystal X-ray diffraction structure, in which we can see the geometry of tin to be octahedric, and we can also see strong interaction between tin and carbon number 1, tin and nitrogen 1, and tin and nitrogen 2. As was compared here in the paper, the length of the bonds are shorter than the sum of the van der Waals radii. Next, for compound number 3, the organometallic 1, we also expected 11 signals. We measured, for this we measured in fluorophore. Even though it's low solubility in fluorophore, in DMSO the 6 decomposes. So we couldn't perform that measurement. But in fluorophore we could see also 11 signals, doublet and triplet. All of them the aromatic regions between 7.5 and 9.2. Next, for tellurium NMR, as I said, it was very low soluble in chloroform. That's why the singlet is not that visible, but it corresponds, it has a chemical shift at around 1270. As an impact, we synthesize more NNC organometallic complexes. These two are just some examples. For the others, for some of them, we could also obtain crystals and were measured. And generally in our group, there is a method to try radiation and then adding the metallic chloride. However, for pyridinic rings, we cannot do that because the pyridinic rings have an electrophilic character. And when we mix it with N-Bully, we will have a nucleophilic addition at the alpha carbon. And we won't have any substitution with hydrogen. So we have to find another way and to probe if that way can work for different metals. And they work, apparently. And it can be used for different transmetallations to group 14, 15, and 16 metals. As a future prospect, from the organometallic compound, we can form an organoiodide. It's a very simple reaction. It doesn't undergo under argon. It's under oxygen. It works very simple. And from this, we can try a substitution with N-Bully. And then try low valence chloride, such as lead dichloride or tin dichloride, because when we tried with lead dichloride, the second transmethylation, it didn't work at all. And when we tried with tin-2, the transmethylation, we could obtain a tin-4 compound. I didn't explain myself why. And as acknowledgements, first I want to thank PhD student Rafael Fontana for all the help in the last almost 3 years in this research. I want to thank Professor Dr. Cristian Silvestru for all the support, and Associate Professor Albert Sorana for the measurements on the single crystal X-ray structures, and also all of the team from Organometallic Chemistry Group in our faculty for all of their help and all of the answers for my questions. Thank you.
Congratulations, Razvan. We can now accommodate a few questions. Yes, from the back.
Although I think it's related to your past work, you were saying that you made organometallic compounds with metal in high oxidation state. But for antimony, it's not really the highest oxidation state.
It's not really, I know.
But were you not able to obtain organometallic tetrachloride, or you haven't tried it?
I haven't tried it yet. It was on my plan to do it after this conference that I might work with Organometallic 5. But we didn't try it yet. We tried only with antimony trichloride until now.
Thank you.
Okay, are there any other questions from the audience? If not, congratulations again, Razvan.
The next presenter will be Claudia Bârcea with homoleptic diorganotellurium-2 derivatives containing pyrazole rings, synthesis and structural characterization.
Thank you for your introduction. Good afternoon. My name is Claudia, and today Sorry. Today I would like to present our latest results regarding molecular derivatives. But before I get to the scientific and structural characterization, I would like to do a short introduction. Tellurium is a metalloid from the carbon family. Organocarbon compounds have uses in coordination chemistry as ligands towards different transition metals. Those compounds being later on used in different applications such as catalytic applications, material science, and chemistry. Today I will present the scientific of tellurium compound. We obtain the tellurium compound by reacting element. Tellurium with in a first step with sodium borohydrate in I'm sorry, I will take a short break. Okay, so I will try again. We react the element of tellurium with sodium borohydrate in sodium hydroxide water and increasing with 40 hours. In the second step, we react with the corresponding bromide, reduce for another 2 hours, and again we obtain our compound as a solid of orange coloration in a very good yield of 87%. The compound was structurally characterized by the NMR spectroscopy and mass spectrometry and used as a ligand in reaction with different salts, with silver salts, palladium and platinum salts. By reacting with the silver trifluoride in 1:1 ratio, we obtain complex 1. In a 2:1 molar ratio, we obtain the complex 2. And by reacting with the palladium and platinum salts in the mentioned reaction condition on the slide, we obtain complex 3 and 4. The NMR spectra of proton and tellurium conform the expected structure for ligand. As we can see in the aliphatic region appear the protons from the NMR degrees at around 4 ppm and 2 ppm. In the aromatic region, we can see the protons corresponding to the protons on the pyridine group. Proton 2 at around 60 ppm and proton 3 and 1 at 7 ppm. The tellurium signals appear as singlet at around 260 ppm. This confirms the formation of only one species. The NMR proton spectra of the silver compound was this the expected one, as we can see while comparing it with the ligand spectra. There was a slight intervention in the experiments. In the resonance signals down periods shift, chemical shift. We also obtained the NMR spectra for nitrochloride and 125 for context 1. As you can see in the fluorine NMR spectra, we have only one signal at minus 78 ppm. These two confirming that we only obtained one species from the triplets ion in the structure. In the tellurium NMR spectra, we obtained again one signal, one singlet signal at around 159 ppm. Here we also have compared the spectral complex Q, as in the spectral complex 1, there was a slight chemical shift in the resonance signals, slight downfield chemical shift, and in this case as well, we obtained only one singlet signal for the fluoride in around minus 78 ppm, corresponding to the triplet anion. In the tellurium NMR spectra, we obtained one singlet signal compared with the signal we obtained for the ligand. This one is situated at 182 ppm. We were also able to obtain suitable crystals for X-ray diffraction studies, as seen in this presentation. The two nitrogen corresponding to the metal center. The metal center after the studies done on the crystals, we could confirm the number 4 coordination, that the metal center has coordination number 4 and the square planar geometry, as you can see. In the study, we could also determine direct association between the peptides in the solid state by being formed by the bonds between the chlorine and tellurium atoms. With the crystallization water, we also obtain this polymeric chain. We were also able to obtain single crystals for the platinum complex 4. In this case as well, the coordination number is 4, and as you can see, the nitrogen also coordinates to the platinum center and confirm the square planar geometry. Here also we obtain, we can see that we obtain polymeric chain by the interaction between the tellurium and the iodine atoms from the molecular Molecular Association. In the end, we were able to obtain a novel and stable homolecular compound containing gravity groups. And we specifically characterized and used it as a ligand for the preparation of simple palatic and metal complexes. We were also able to obtain the suitable crystals for complexes 3 and 4 and fully characterize all of our compounds. In the end, I would like to thank our group for all the support during the sciences and during this week for everything, for the competitions and help with my score that it was not very good, but next time. And of course for financial support and you for your attention.
Claudia, it was a great presentation, don't worry. Now, do you have questions?
Yes. Thank you for your presentation and congratulations on your work. I have more of curiosity. Which one of your compounds was the most challenging to obtain?
I think, in case of the silver toxicity, it was a bit difficult as we needed to work in, I don't know how to formulate it right now. We need to work with the silver complexes in a dark environment. They're more sensitive to the light. So this was a bit of a challenge. And of course, with their preparation after purifying process and their I was so scared.
Okay, are there other questions from the audience? No? In that case, thank you very much once again.
Now, for our last presentation today, I will invite up front Nicoleta Stoian, who will present us the chemical structure and target space profiling, a layered SOM approach.
Thank you. Hello, my name is Nicoleta Stoian. I come from Western University of Timișoara and I work at Marion Andreevulescu Institute of Chemistry. Today I'll present my study in chemical structure and target space profiling with a self-organizing map approach. Our motivation was that current drug discovery methods use mainly chemical profiling. We know a lot about how from the structure we can assess toxicity or bioactivity, but we know less about biological profiling. It is less accessible, but nevertheless important. So I wanted to create somehow a complex overview of drug and their target variants using unsupervised learning. What is a self-organizing map? This is what it looks like. You can see different colors. Those are regions, and regions have common properties. You can see those circles. Those circles are region units. We call them neurons. And inside them there are dots that are data points. Those data points in our case represent drugs. So we have grid size, epochs, which are the number of times the entire data set passes through the algorithm, layer weights, and learning step, why we picked self-organizing maps. They are multi-layer compatible, so we could have a chemical layer and a biological one. First, our dataset. The dataset was from Drug Central database. It consists of 2,500 drugs. We filtered them to be approved drugs, to be organic compounds, ideally small molecule, and to have either activity value or known mechanism of action. And since Drug Central also has veterinary use drugs, we only pick the ones who also have human use. In the end, we ended up with 1,600 drugs, 2,600 biological targets, and 21,200 unique drug protein pairings. How we encoded the data, I picked the max fingerprints. It is like a binary string of 166 questions you answer based on the structure. And then our real challenge was when we tried to encode amino acid sequences from the proteins to have numerical data. And I found this Prodbert tool. It is like masked language model. It takes solely amino acid sequences and turns them into something like this. The vector is longer. It has 1,024 units. So we had two cases. We had a drug with one unique target, so it was pretty straightforward, but we had to somehow create a single vector if the drug had multiple targets. And we performed multiple normalizations, and we ended up with a matrix. After that, as I said earlier, you can customize your self-organizing map on grid size, epoch, and so on and so on. So we ended up with almost 2,000 models, so we have to discriminate somehow. And we picked a quantization error and topographic error thresholds. A low quantization error means that data points gather inside the neuron, and the high quantization error means that they somehow want to migrate towards a different neuron. For a low topographic error, the neurons that are similar stick together. So with a high TE, the map is more scattered in properties. So we ended up with 243 models. We variated the weight, meaning if the chemical layer is more important or the biological one is more important. And we found out that we have better error values for 1 to 2 weight in favor to the biological layer. But 243 models is still a lot. So we saw that a lot of our models had a lot of empty neurons, and we wanted to have another discriminating criteria. So we said a percentage I'm sorry, an empty neuron percentage threshold. So we ended up with 21 models, and we performed hierarchical clustering to see how well the regions split. We wanted to see how well they split at 6 regions. After that, we ended up with one single model. This is the parameters, and we'll represent data like this. We have points and their properties as a pie chart. For region 1, this is the region 1. We can see that a lot of GPCR1 receptors are here, and a lot of protein kinase active drugs are clustered in region 1. This is like a large region and also diverse in biological properties. For region 2, we have those neurons which are interesting because they have more diverse percentages of enzyme active drugs. And we also found out that those drugs are also more active on bacterium and viruses. For region 3, just like region 1, it is a large region, so it is logical that it is diverse. So we saw that the mapping of enzymatic drugs have like an aggregation here of prostaglandins synthetases and glycosidases, and a lot of transporters are in this region. Oops. For region 4, we figured that since it is in the middle of the map, it is more structurally diverse, so we performed Tanimoto. We also took a step back, and we saw that the entirety of region 4 derives from region 1. And you can look here, for region 1, the mean Tanimoto is 0.5, for 4 is 0.374, and if they are together, they have 0.431, which confirms that they are structurally diverse. For region 5, we saw that the scaffolds resemble a lot the endogenous hormones, and it is pretty logical that they are active on nuclear hormone receptors. And for region 6, we saw functional aggregation of sulfonamides. We looked for sulfur atoms count in drugs per cluster, and we saw that a lot of drugs in region 6 have at least one sulfur atom. And these are the functional, the common uses of drugs in cluster 6, anti-neoplastic being the most common one. In conclusion, it is a powerful framework for chemical biological space exploration. It could be used in the future, maybe, when I fine-tune it, for drug repurposing and early target assessments for drug candidates, in silico profiling, and polypharmacological analysis. Thank you.
Thank you, Nicoletta. Now, I guess start from that.
I didn't hear you. With other existing software? No, I did not. I did not see anybody using Prodbert. I did not see other methods starting from amino acid sequence and using it like in a machine learning way. If I instead of Drug Central, yeah, of course, I want to do that in the future too. But Drug Central was more accessible to me when I designed it. What do you mean? From 1600 to 2600 targets. 21,000, yeah. Because before that, I filtered to have activity values strictly on that target and known or either known mechanism of action. So a lot of them were thrown out. Like the maximum I had before that threshold was around 60,000.
Thank you again.
Thank you.
Okay, now we're coming to the end. What a day this was. Stunning presentations, delightful treats, savory coffee, and a brilliant vibe overall. We are grateful for everyone who was here today, and we hope you will make it to what is left tomorrow here at the conference. But to the participants, don't think that today ends here. You are expected to take part in the second social activity of the week, starting at 8 p.m. at GAP. To everyone else, Have a wonderful afternoon.